Our next speaker is Riddha Manna from all the way from Switzerland, EPFL, and he's going to talk about robots. Oh, that's what I'm going to say. Automated cobot-assisted high-throughput phenotyping of cognition in behaving Drosophila melanogaster.
Hello, everyone. So today I'm going to talk about my PhD project, which is to develop an automated high-throughput system for phenotyping cognition in behaving the Drosophila melanogaster. So we define cognition as the ability to acquire information, retain it, and apply it to adaptive behavior through information processing. Now, we have been studying the fly, which shows all the four components of cognition for decades. So to study fly cognition and behavior, any that type of systems have been developed, the majority of which can be of two types. The TMS setup, which is excellent for studying mainly olfactory learning, but they only measure group behavior. And the other type is the treadmill or the airball setup, which can be used to study a single fly at a time in great detail. There have also been many high throughput technologies. For example, the maple from Harvard, which can actually score hundreds of flies, but that does not implement any learning assay. And there are also algorithms for highly accurate ethomics, and there are setups to implement associative learning, but they have not reported any kind of any automated fly handling, right? So we want to approach the problem of implementing visual associative learning in flies in a high throughput manner with two strategies. That is to scale up the number of flies per experiment, and as well as to automate the experimental setup to increase the number of experiments in a day, right? So to scale up, we are using Y mazes. We project colors to the fly using an LCD screen. And on top of that, we have an electrified transparent grid to shock the flies using an associative learning assay. And with the design of our mazes, we can track the flies' behavior and decisions. So each maze is about 3 centimeters in size, and on each glass plate we have 8 such mazes. And at a time, our setup is able to handle 8 such plates, so increasing the throughput to 64 flies per experiment, right? And all the parts are made so that they can be handled by a robotic arm, and the glasses even have holes in them for anesthetizing the flies inside the mazes for retrieving them after the assay, right? So we are controlling the colors as well as the shock for each individual maze through a closed loop control, and have a real-time analysis of the flies tracks, velocities, their decisions, as well as the learning score. And there is no need of a post-hoc video analysis, right? So to validate our system, we implemented an operant conditioning assay where one of the 3 arms was associated with a color, and the fly was hooked on that particular arm. A wild type fly actively avoids the shocked arm throughout the experiment, which is actually apparent in these traces where you can see this is the time axis and it's the X and Y represented by like the X and Y and phi on the measures. When it is not shocked, you can see that it goes in all of the arena, while if it is shocked on the top arm, it actually avoids that arm. And yeah. So so we measured the fraction of the total time a fly spent on the soft arm. We compared the wild type fly with memory and blind mutants, which cannot associate the shock with the color. And we see no significant difference between the shocked and the non-shocked groups for the mutants, while the wild type shocked group actively avoids the shocked arm. So far, we have screened 37 wild type genotypes from the Drosophila genetic reference panel with around 2,500 flies, making 230,000 plus decisions. And we see that 19 of the 37 wild type genotypes show significant learning. And just for the assay, it took us just 1 week to get the results. And of course, it does not include the fly pairing steps, right? So we can track the learning scores throughout the experiment for each individual fly. We see for the non-shocked groups, the flies showed a tandem behavior, while for the shocked groups, they show a well-directed behavior converging to its final learning score. Now, to make things faster, we are developing an automation protocol. Using collaborative robotics, we can automatize the setup of the experiments. So these robots are easy to implement. They have human-like arm manipulation, which enables us to implement already established fly protocols, and fly manipulation is not restricted to 2 dimensions, unlike like stepper or servo control rotor-like manipulators. So finally, the robot actually needs to anesthetize, identify, place, and track the flies automatically during the experiment, as well as retrieve the target flies after the assay, right? So using a 2-arm collaborative robot, we can actually set up an experiment in under 19 minutes. It is not only lesser than the manual timing, but it will also mean a more uniform handling of the flies across the replicates, which would reduce the variation due to handling, and we can actually capture the biological variability in behavior. So with the fully developed protocol, we intend to screen 1,500 flies in a day, right? Yeah. So I'll have the video play for a bit so that you can see the steps, right? Yep. And with that, I want to thank my supervisor, Anna, and our collaborator, Tomislav, who is from the University of Zagreb, and our lab members, Ivan and Samuel. I would also like to thank the BrainMind Institute at EPFL and the LSE funding. Thank you for your attention, and I would like to invite you all to come have a chat with us for any kind of a collaboration. And if you have any cool ideas about how we can utilize or improve our system. Thank you.
Thank you.
Yeah.
Just like, I'm gonna step. So how much?
Hmm?
How much is the robot?
Oh, it's
Are you
around
taking
50,000.
orders? 50,000?
Yeah,
Oh, you're not gonna ask how much question?
around 50 to 60,000.
Well,
Yeah.
I think it's an awesome system because we've been always, Oz is there and we've been always thinking about high throughput to do behavior. So I think this use of this robot is really cool. One thing that I was interested was your, so conditioning, kind of fear conditioning stuff is light-based. And you
Yeah.
have already kind of hits from your GDP, just like a reference
Yeah.
panel where, it's learning well and learning, not well.
Not well.
Have you retested those lines in an olfactory based, old fashioned style T-maze thing?
No,
And
not
does
yet.
that reproduce your data?
Not yet, but we do intend to do that to see.
Great, thank you.
But I would not expect their olfactory performance to actually match with their visual performance.
Yeah, but I think
Yeah.
if you still find something that's only affecting olfactory based one, but not the vision one, for example, or the other way around, you find something with the vision, it affects it, but then if you test it with olfaction, it doesn't. Then you might start seeing some different kind of genes that are specific to senses.
Yeah,
So,
yeah, of course.
thank
Yeah,
you.
Thanks.
Okay. Thank you, Riddha. Amazing.
Thank you so much.
Okay.